Hello students. Welcome to my channel. Our today's topic is plasma membrane. So, let's get started. First question is, what makes up cell membranes? Cells are wrapped in a special skin called the plasma membrane. It's like the outer wall of a house, connected to inside walls, cytomembrane system, that divide the cell's gooey filling, cytoplasm, and hug the nucleus, the cell's control center. All these membranes are built from lipids, fats like phospholipids, cholesterol, and glycolipids, and proteins, mixed roughly 50-50. Lipids stack into a lipid bilayer. Think 2 layers of soap bubbles stuck together, about 2 molecules thick. The water-hating, hydrophobic, tails hide inside, away from water, while the water-loving, hydrophilic, heads face out toward watery areas inside and outside the cell. Proteins float or sit inside this bilayer like boats on a wavy sea. It's called the fluid mosaic model, mosaic because it's a patchwork. Some proteins punch all the way through, transmembrane proteins, thanks to their water-hating parts. Others just stick lightly to the surface via fat tags. These are all integral proteins stuck tight. Peripheral proteins are looser, just hanging on to other proteins. Sugars, oligosaccharides and polysaccharides, attached to proteins, glycoproteins, or lipids, glycolipids, and mostly stick out into the space outside the cell, like flag signaling, this way for cell talks. Lipids aren't evenly spread, they clump into tiny zones called lipid rafts, detergent-resistant membranes, packed with sphingomyelin and cholesterol. Certain proteins love these rafts, which supercharges jobs like immune cell signaling, T-cell receptors, or cell gluing. It's like VIP lounges in a club for special teamwork. Second question, what membranes look like under a microscope? In electron microscopes, fixed membranes, stained with heavy metals like osmium tetroxide, show as 2 dark lines sandwiching a clear middle. The unit membrane, total 7.5 nm thick per layer, or 15 nanometers for plasma membranes. Freeze fracture splits the bilayer along its greasy middle, like cracking an Oreo cookie. Each half has a surface, facing outside or inside the cell, and a face, the split side. Outside face equals extracellular fracture face, by exoplasmic or extracellular surface. Inside equals protoplasmic fracture face, by protoplasmic or cytoplasmic surface. This reveals intramembranous particles, big proteins or protein teams embedded unevenly, more on the inside layer in plasma membranes. Clumps often form gap junctions, tunnels letting ions or molecules zip between cells. Third question is, how membranes move and work. Biophysics proves the bilayer is superfluid, like olive oil. Proteins slide around unless tied down inside the cell. Membranes act as smart gates. They block random stuff, creating separate zones for life's jobs, like kitchen versus bedroom in a house. Example, imagine the membrane as a city fence with security guards, proteins. It lets friends, nutrients, in but keeps strangers, toxins out, while traffic, diffusion, flows smoothly inside. Thanks for watching. Please do subscribe this channel and press the bell icon.